day, learners! Welcome back to my channel. For today's lesson, I will be discussing the second stage in cell cycle, which is about cell division. Particularly, it focuses in mitosis. Have you ever wondered why organisms change? As time goes by, every organism is changing. Aging is one of the common changes in different organisms like us, human, simply because the cell divides and matured. Cell division happens in an organelle of the cell called a centriole. It is responsible for organizing chromosomes before cell division. Our learning objectives in this lesson: compare mitosis and meiosis and their role in the cell division, and explain stages of mitosis. Do you think will we run out of skin someday if we lose thousands of skin cells when rubbing our hands or when we get wounded? The answer is no. Why? Simply because new cells are formed to replace dead cells by the process of cell division. What functions does cell division play in our body? Aside from replacing dead cells, cell division is also a way to produce new tissues during growth and development and production of new generations of individuals among some groups of organisms. We have two kinds of cell division. The first one is mitosis. Mitosis is a type of cell division where multicellular organisms undergo growth and development and when they repair damaged tissues. The other one is meiosis. It is a type of cell division used to produce sex cells such as egg cell and sperm cells. M phase or stages is divided into two. We have mitosis and meiosis. Mitosis occur in body cells. It has one division per cycle, and it produces two genetically identical cells or known as diploid cells. While in meiosis, it occurs in sex cells. It has two divisions per cycle, and it forms four haploid cells. Mitosis composed of four stages, namely prophase, metaphase, anaphase, and telophase. It is commonly known in abbreviation called PMAT. After interphase, the genetic material is actually unpaired, uncoiled, and long thin structure composed of DNA and protein called chromatin. During the early stage of mitosis, chromatin becomes chromosomes because the uncoiled chromatin is folded and condenses into characteristic formation called chromosomes until it becomes visible and serves as a guide in the process of mitosis. During prophase, the long and thread-like chromatids during interphase starts to coil, the nuclear membrane disappear, spindle fibers are formed, and centrioles migrate at the opposite poles of the cell. After prophase is the metaphase stage, wherein chromosomes move and align themselves at the center of the cell called metaphase plate, or in other words, equatorial plate. Spindle fibers connect each chromosome on its centromere, particularly in kinetochore, to the centrioles located at the opposite poles. For us to be able to understand what kinetochore is, let us describe each part of the chromosomes. Sister chromatids are the identical copies formed by DNA replication. These are commonly the sides of the chromosomes, and it is identical because it is replicated during the interphase stage. The next one is the centromeric region, or known as centromeres. It links a pair of sister chromatids. It is located on the central part that attaches the sister chromatid. The next one is the kinetochore. Kinetochore attach chromosomes to spindle fiber. It is very important part of the process during mitosis. And lastly, the mitotic spindle microtubules, or known as spindle fiber. It is responsible for moving and segregating the chromosomes. The next stage of mitosis is anaphase. During anaphase, the sister chromatids of each chromosome divide or split and move toward the opposite poles due to the shortening of spindle fiber. The 4 double-stranded chromosomes during metaphase become 8 single-stranded chromosomes because they migrated in opposite poles. The number of chromosomes depends on the kind of organism. The last phase of mitosis is known as telophase. During telophase, the chromosomes are located on opposite poles and the spindle fiber disappear. At the poles, the chromosomes uncoil and the nucleolus and nuclear membrane begin to reform. That's why this phase is also known as the reverse prophase. After telophase, cytokinesis occur. Cytokinesis divide the cytoplasm of the parental cell into 2 daughter cells. The daughter cell have the same number of chromosomes. So those are the stages of mitosis. Again, the stages of mitosis are prophase, metaphase, anaphase, and telophase. As I mentioned earlier, people get aged and die. But why other people look so young and live longer? Is there any secret in immortality? Science believes that the secrets of looking young and living longer is in our chromosomes called telomeres. Telomere is a special protective cap located on the edge of chromosomes. In every cell division, the telomeres get shorter. That's why, as the organism is aging, the telomeres gets even shorter, that the cells stop dividing and lead to shorter life. Telomerase is an enzyme found in telomeres responsible in elongating chromosomes. It acts to delay the shortening of telomeres. Science is still continue conducting different studies in preserving youth and keeping body cells actively. To sum up our lesson, the cell cycle is divided into 3 stages: the interphase, where the cell grows, DNA gets copied or replicated, and it is the preparatory stage for cell division. The next one is the mitosis, consists of 4 stages. We have prophase, metaphase, anaphase, and telophase. And last but not the least, the cytokinesis, where the cytoplasm is divided. To check your understanding about the lesson, it's quiz time! And that's all our lesson for today. Thank you, grade 8 students. I hope you learned something. If you want more videos, don't forget to like, share, subscribe, and turn on the bell for more updates in our science class.